For more than 2 years of a global pandemic, those who weren't so sure about taking COVID-19 seriously would say, but it's just the flu. That same flu, the World Health Organization says, causes severe illness in 3 to 5 million people every year, killing up to 650,000. After lockdowns and social distancing, anyone in a workplace or with a kid in school will tell you the flu is back. And while vaccines do work a little just for the current flu season, there are those, like our guest today, who are trying to go a step further and develop a universal flu vaccine. This is the InnerView. And that company you've just seen on Netflix has been funded to the tune of $10 million. Dr. Jacob Glanville is the CEO and president of Centivax. He joins us from San Francisco. Jake, good to talk to you once again. First of all, congratulations. Tell me what the $10 million means to you and your organization and what you can do with it.
Sure. Well, thank you for having me on, and thanks for the congratulations. So the $10 million is extremely helpful for the stage that we're at. It enables us to initiate manufacturing of cell lines, which we begin in January for our universal flu vaccine. It also enables us to run some additional experiments. We believe that our vaccine also inhibits transmission. This is something that often isn't tested for vaccines. The focus is on making sure the person doesn't get sick or gets less sick, but we believe that we also stop it from transmitting. And so we're testing that in preclinical models. There's also some questions around the what's called original antigenic sin, or immune imprinting, where being exposed to historical influenza strains will sometimes change unfavorably your response to new vaccines. Our vaccine's been designed to fix that problem, and we're confirming doing additional experiments in preclinical animal models for that purpose.
For those of us who read the news about the study that came out of the University of Pennsylvania, published in the journal Science, where they said that good news regarding a possible universal flu vaccine, mRNA, they used 20 old strains of the flu, somehow, put it together, and it seemed to work well in animal studies. I'm assuming your model's a bit different. Tell me what's the difference?
Sure. So what they demonstrated is that you can mix together multiple different RNA sequences for different hemagglutinin spike protein strains, and that the animals produce an immune response against each one. What they also showed, though, it didn't really produce broad immunity. So they put one strain for H1N1, one for H3N2, and one for a number of other of these subtypes of influenza, most of which do not infect humans regularly. But when you look at within H1N1, they only really protected against the strain they put in. It didn't spread to other more distant strains. And that's really the whole problem. You want a vaccine that protects not just against that year's H1N1, but very broadly. And their technology did not do that. That's the area that we focus on, because in the end, humans are almost entirely affected only by H1N1s, H3N2s, and HABs. And so the majority of the human problem is to have one shot that provides you broad anticipatory protection across those three subtypes.
When I read that abstract, it also said that the mice and the ferrets that they tested on also showed a really good immune response to an avian flu of sorts, some sort of bird flu, right? So again, to me, a layman, this sounds really good, and it sounds as if this thing toughens them up for all manner of viruses. Tell me what's going on there.
Sure. So look, first off, we like that study because that study demonstrated to us that our technology would be compatible with RNA. What they showed is of the 20 strains they put in, 17 of them were strains that mostly affect birds. Sometimes, like H5N1, those can cross into humans. They can be quite deadly, but they don't really transmit very efficiently. So they demonstrated that they could hit the 20 different subtypes, but they only show that it's effective against one representative strain. So the way to think about it is you get the shot, you'd be protected against all the subtypes, 17 bird subtypes and 3 human ones, but really only for that season and a couple seasons around it. So in that way, it's not actually that different than a current flu shot.
Tell me why it's so difficult to actually develop a universal flu vaccine. Let me ask this, giving you a little example of myself getting a flu shot a year ago in Cape Town, right? So, or a year and a half ago in Cape Town. So it was June, and it's the winter in South Africa. And as I'm getting the flu shot, I'm thinking, I'm not so sure if this is from 6 months ago in the Northern Hemisphere, it's their old flu shot from their winter, or if this has been rolled out for us in the Southern Hemisphere before everybody else, and this is the guesswork that's been done where they're assuming these are the strands that are going to go out around the world, and we're getting it 5 or 6 months before everybody else. But let me just, I don't know, better safe than sorry. Let me take my chances. So, I mean, I assumed that it was going to be more helpful than harmful, but was my skepticism vindicated in some sense that it is guesswork, and this isn't something that is as solid as we'd want it to be in a seasonal sense?
Yeah, so I think you got it right. I'll give you a little background on that guesswork, and then I'll tell you why it's been difficult to create broad spectrum vaccines so far. So the guesswork really dates back to the 1930s. That's when we started creating vaccines for flu. But unlike some other pathogens where it's a one and done and you make a vaccine and then that's just what we give people for decades afterwards, flu mutates, and it mutates pretty quickly. And so we have been stuck in this Sisyphusian pattern for decades where every year they have to update the flu vaccine because it's deteriorated. It's no longer as effective against the new strains of flu. This is analogous to what's happening with the coronavirus now and updating that vaccine. And it's a balanced risk profile. They have to guess a little bit because you need time to manufacture a vaccine. And so what people do is it's the CDC, the World Health Organization, and a couple other groups that work together to make decisions about this, and they release a guidance. They release a separate guidance in the Northern and Southern Hemisphere, and they say, hey, folks, here's what you got to put in your vaccine this year, guessing what the world's going to be like 6, 7, 8, 9 months in the future. And they kind of are fixed with that. It's actually a little worse than what I just mentioned because they also know if they gave out 4 new strains that you had to manufacture, each manufacturing bears a risk. And so they make a compromise where they say, you know what, we're going to let you keep some strains from 2, even 3 years ago in there because maybe they'll be good enough, and we want to balance updating from risking that there's going to be manufacturing delays and then we don't have enough vaccine. And the consequence of all that is that your vaccines are somewhere in the 30 to 60% efficacious range, like effectiveness for flu, which is not great. And in the last 5 years, it's been more on the low end of that range, sometimes as low as 10%. And so that's you're thinking about it's right. When I take a flu vaccine, I'm taking it because I say it's increasing my odds of not having to deal with this, but it's not particularly effective. It's just the best that we've got. To your second question, why has it been so hard? So we've known since, I believe the first paper was in 1993. There was a research group that identified, hey, look at this. Someone has created an antibody that isn't just hitting one strain. It seems like it's crossing diverse strains. And then more papers like that started coming out in 2000, 2004, 2006. What they were realizing is that although the virus mutates every year, there are certain sites, these Achilles heels on the viruses, that the virus is unable to mutate or it's no longer infectious. And if you can make antibodies that specifically target those sites, then those antibodies provide broad protection against wide panels of influenza strains. And that got everyone very excited in the vaccine field because they said, well, if we can just get our vaccine to make those kinds of antibodies, then we will have a universal vaccine. And it's turned out it's been challenging to do that in the first 20 years of efforts. The problem first was that the immune system was a very complex black box. We didn't really understand what was happening inside that system. We would put a vaccine in and the wrong antibodies would come out. And so people tried a few things, but there was absent engineering theory on how to force the immune system to do what we want. In the last 10 years, there's been major advances in high throughput immune system interrogation technology. So deep sequencing instruments applied to the immune system, high throughput laser-based cell sorting assays, mass cytometry. And these have given us additional guidances over why the immune system is so hard to focus on these sites we really like. And for that reason, we have a meaningful advance in this space, and there's competition. There's other groups, and the race is now on to create universal vaccines. And I think that there's meaningful progress by multiple groups that are coming out now. This is a result that's going to not only affect the flu, which is great to have a single shot that actually works reliably all the time, but it also bleeds over into broad spectrum coronavirus vaccines, progress towards a working HIV vaccine, and a number of other rapidly mutating pathogens.
Yeah, that's an interesting point that you make. So HIV as well, coronavirus as well. And I wonder, in those 2 years or so, maybe slightly longer than 2 years, did we underestimate the flu while we collectively, globally tried to tackle COVID?
Well, yeah, it's certainly COVID sucked the oxygen out of the room. It wasn't all bad. There was this interesting side effect of the coronavirus pandemic where, because of all the masks and the isolation, we had 2 very benign flu seasons, and there was one of the 4 strains that we track apparently went extinct. So they're one of the HIV strains we haven't seen in 2 years.
Amazing.
That said, we're having a pretty gnarly flu season now, and partially that's because everyone's coming back out, and there was less exposure to the virus, there was less vaccination, and now we're paying the price for it.
And when we look at the success of developing vaccines in record time, I mean, we chatted when the endeavor had just begun. You were hopeful, and I guess you were very much vindicated that everybody who got together in trying to produce these vaccines could do it, and they did it. They did it in, 11 months, in less than a year, when the guidance is ostensibly this takes 5 years, 10 years, whatever. Does that mean that this is translatable into developing new vaccines for the flu, a universal vaccine for the flu, for HIV, and so on? Or is it only something that can be done in the context of a global pandemic where everybody switches on and says we don't want mass deaths?
So it's a bit of both. Certainly there were some acceleration capabilities that were made, decisions by vaccine producing companies, but also policy changes to accelerate vaccine production by the FDA and regulatory authorities. And some of those tools have relaxed back to normalcy. But there were also meaningful advances that we will continue to benefit from. One is that the RNA technology clearly has a manufacturing preparation advantage. They can manufacture an RNA vaccine faster than you can manufacture any other vaccine. The other things are really honestly are a mindset issue. I think that there is this tendency where people look to the past for how long things take, and they assume it's going to be that same way going forward, but that is not true in a space where there's technological innovation. And I think there is a greater expectation now in vaccine science that it is possible to accelerate your vaccine programs faster than has historically been possible. There's fundamental limits. You have to have safety. You need to be able to assess for toxicity. You have to be able to satisfy a set of regulatory guidelines by the FDA and other agencies globally. And those are for good reasons. You want to make sure people are safe and the vaccine actually works. But that said, I think, yeah, we are looking at a faster turnaround time now for new vaccines coming online than were historically possible.
Those University of Pennsylvania scientists, biologists, they had used mice and ferrets as their subjects. As far as I understand, you use pigs and ferrets as well, right? As biologically and genetically similar as they are to us, there's still a leap then to go to human trials. Tell me what your thought process is when it eventually gets to human trials, and what you expect to happen, and some of the possible contingencies you have in mind, and maybe some surprises you might have in mind as well.
Sure. So we do what we can before we go in. In some ways, the immune systems are similar, but the more, as an immunologist, I look into every organism, the more they have like a weird, bizarre, different immune system story to tell. And for that reason, we test on multiple different animals before we go to humans. And then even in humans, there can be some surprises. So I chose to work on pigs initially because they actually get the flu, and it's a $180 million per year market for flu shots for pigs, and they have the same problems humans do of mutating viruses making their flu shots not very effective. Ferrets are important because they have lung tissue that's very similar to humans, and it's become a regulatory precedent. People have said, well, lots of people have tested flu shots on ferrets, therefore we'd like you to test them on it as well. You do what you can, you get everything ready to go, but ultimately you have to go into humans. And there's a couple things we can probably expect based on the other animals that have been worked on, but there could be some surprises. Humans live a lot longer than those other animals. Those experiments typically run for, the equivalent of like 1 season, 1 year at most, where a human has been around, they could be around for 70 years, and they could be exposed to generations of influenza viruses and vaccines. And that's why we spend time looking at this original antigenic sin or imprinting question. The other thing that happens with a human is because they have such a long lifetime, as people get above 55 to 60, this immune tiredness called immunosenescence begins to kick in. And so it's a new problem. They have, on one hand, more immune memory. There were people that had immunity back to 1918 they were detecting during the 2009 swine flu pandemic. So there's this amazing ability of your body to store immune memory, but it's harder to get the cells to get them active. And so there's unique considerations with the elderly on applying adjuvants. So these are immune annoying chemicals you put into your vaccines to make the immune system wake up or give them higher doses. So those are some unique considerations. I think what I would expect to see is the same thing we've seen in our ferrets and our pigs, which is ultra broad spectrum, high titer immune responses. I expect them to go out for months, as we've seen with those other animals, but the questions that I have that I don't have a basis for is how much beyond that 9 months, and that's what we're going to have to track these people for to understand. It produces this ultra broad response. How long can they go before they should get a booster shot? Is it 1 year, 2 years, up to 5 years, as we have with tetanus shots? That I just don't know until we start looking at humans.
Safety is a concern for a lot of people. Over the 2.5 years, this became vaccines became a depth charge in culture wars. People worried that there might be unintended consequences, or sometimes for the conspiracy theorists, intended consequences from those giving you the vaccine. But sometimes I feel like, you do have some people with a genuine curiosity who are worried about safety, and their voices get drowned out amid the two camps in the culture wars. Somebody might be genuinely asking some questions about safety and efficacy, but they're called vaccine deniers and the like. So I guess for clarity and to set the record straight, when it comes to safety and when it comes to possible side effects, when you're developing a universal flu vaccine, what do you worry about and what are you certain about?
Sure. So I can agree with everything you just said. I struggled during the pandemic to communicate, yes, these vaccines are good and safe, and yet we also had an obligation to announce that in rare cases there was myocarditis and pericarditis. And it was a challenge to communicate that because some folks were saying, it's problematic to even share that information too much because it'll freak people out. But I felt that I had to say, yes, it's safe and it's worth it, but there are these rare cases, you should be aware of it, particularly the ages 12 to 17. And I think that we owe, as epidemiologists, a responsibility to the public to try to be as transparent as possible, to not try to overgame the system and just say, look, here's what we know, here's what we don't. I still think this is why I'm vaccinating my family, and I'd recommend that you do the same for yours. So that's the challenge we'll continue to have. With respect to influenza, you need a good safety profile. We've been vaccinating people since the 30s. Almost all of that vaccine history has been quite safe. And typically with low number of side effects. So people are used to taking a flu shot and not feeling super knocked out afterwards. And so I think that's sort of your parameter space that we in Centivax have to work with, because I can't, for instance, use an adjuvant that would produce super awesome immune titers, but would have people feel really knocked out for 3 or 4 days, because people aren't going to take it. They're going to be like, no, I'm used to the flu shot not being annoying. And so that's the criteria. The good news with flu is that it is our most studied vaccine ever, right? Because we make 2 new ones, one in the northern and one in the southern hemisphere, every single year, and we've been doing it for the better part of a century. And so we anticipate great safety because our technology works off of very similar principles with our unique technology to induce breath. But of course, that's something that is essential that we do observe in those studies, and so that's what we look to.
Yeah. You mentioned earlier on that in terms of the flu, colds, regular colds and flus, they had a pretty weak and tepid time during the pandemic because of social distancing, because of lockdowns. But now that they're back, because everybody's back in the workplace, kids are back in school, there's some suggestion that kids are not as equipped as they, particularly kids, are not as equipped as they would normally or ordinarily have been had they been out in the mix catching all the different bugs over those 2 years. Their microbiome is different. They grew up during a pandemic. Could you address that a little bit for me? Are kids less equipped and weaker in the face of something like the flu because of the 2 years that they spent at home?
The way I would think about it is I would remind people that there are people all over this world that have very different lives. You have kids that live in a city. Yeah, being isolated compared to hanging out with lots of other kids in a city means they've been exposed to less pathogens. But like, has life really changed for someone who lives in an agrarian environment, who lives in a small village, who didn't have that much contact? So no, it's so fundamentally what the real question is have people been exposed to less pathogens compared to people who lived in a city and were interacting a lot? And the answer to that is yes. And there's going to be some catch up now that you're moving back into a city. If that's where you live and you're going to be contacting lots of kids, then certainly you're going to have to be, you haven't built up that childhood history of exposure that's going to create you increased resilience against new pathogens that come. And so now there's going to be a big bump of a bunch of pathogens cycling among kids who have more naive immune systems. But fundamentally, are you weaker? No. Like, if you imagine some kid living in a village in Argentina, it's not like he's weak. It's just that if he were to move to a city, yes, he would be exposed to more pathogens, and therefore you'd have to build in that sort of city appropriate immunity. And so it comes down to where you live in the world and how much exposure to different pathogens you have, therefore how much sort of immunity you're going to have to build in. Essentially, there's a hazing process when you live around a lot of other people where you have to get exposed to things and eventually you establish immunity. And that's where we are.
When we began this conversation, I congratulated you on getting that big cash injection so that you can try to do the work that you do in order to hopefully get this universal flu vaccine. I wonder now, after COVID, has it been easier for you to communicate the science to private investors, to government, to the general public, because everybody had to go through this together?
It's an awesome question. So yeah, it's on one hand, I feel like the whole world has just gone through a massive training course in immunology, virology, and vaccinology. There's, before the pandemic, I would struggle to explain to my grandmother what I did, and I don't think she really understood it, where now she calls me up and says, oh, do you think I should delay to take the Moderna or the Pfizer because I see slightly different IgA rates, and it's astonishing. I see it also when
awesome.
I speak with, yeah, with politicians. They clearly, I think it helped that their policy-making decisions are now much more informed by force of having to deal with this. I still do see there's a remarkable head in the sand tendency that I watched through the entire pandemic, and I still see it now, where since month 4, through the entire pandemic, there's groups of people who say, well, this is about to be over and we don't have to worry about it anymore. And I think there's nothing you can really do to convince people. Of course, they want that to be true, but the pandemic is converted to be endemic. It's here to stay. There will be new waves. And moreover, there's more people on the planet than there ever have been before. We use mass farming measures in a way that causes viruses to jump across the animals and into us, as happens with pigs. And if you look at the news, the rate of new outbreaks has increased because we travel faster than we ever have before, and there's more of us around. And so I think that I still don't feel that the reality of the century we're moving into has fully dawned upon the regulators, decision makers, and policyholders. And so I think that remains a challenge that is going to require, unfortunately, additional major outbreaks and hopefully not, but realistically likely additional pandemics before we adapt fully to the reality.
And with that in mind, whether it is influenza or a coronavirus, are we at least a little better prepared for the next big one?
Yes. So the answer there is yes. I think that the difference between this pandemic and probably all the other ones in history is that it came to us when we were at this golden age in biotechnology. We had the full force of the internet and communications there. We a relatively peaceful world with obvious exceptions. It enabled us to bring online new technologies. So the speed with which we brought on these new vaccines, these new medicines was unprecedented. And and it spurred a huge amount of investment in accelerating future additional technologies. There were, beyond the technologies, there was coordination efforts between nations, the attempt to create, that playbook that says, okay, an outbreak's happening. What are the steps that I do? When do I need to do lockdowns? When do I need to do other steps? It's not that everyone will adhere to them, but at least that exists. And it solved simple things like when the RNA vaccines came out, the question was, well, how are we going to distribute something that needs to exist at -80? And they solved that cold chain. There's still holes in that cold chain that need to be dealt with, but I think that there were lots of progress making sure that we have a global network to monitor for the presence of virus, even in our stores, and to register that data globally so we have more sensitive ability to detect any new pathogen as it emerges and be able to get medicine created and then out to the world, as well as vaccines. And I think those things have set us up for better success, and that's a good thing.
Dr. Jacob Glanville, I always learn a whole lot from you, and I'm very grateful that you took the time. Thank you so much.
Thank you for the insightful questions and for offering to have me on, as always.
All the best. Bye-bye.